Okay. See, supernova is something that always happens in larger stars. But sometimes when smaller stars are there, whatever you have learned in the life cycle of star, that is, when you take an average star, the end of the life cycle is white dwarf. And then if you take a massive star, it can either lead to a neutron star or a black hole. This is the static portion. But when they exist in nature, they may interact with other objects that are nearby. So if you take a white dwarf is smaller in size, but it has higher density, higher gravity and everything, correct? So sometimes a nebula may be present here, or maybe some other larger stars may be present nearby. The star may be larger in size, which means the mass may be still lesser, or the density may be lesser. Then this black hole, this white dwarf, it starts taking matter from here, which means for this, the mass reduces. But for white dwarf, the mass increases. But there should be upper limit for this, no? The upper limit is 1.4 times the solar mass. After 1.4 times the solar mass, it cannot remain stable. Then it will detonate. Ideally, it should be called as a supernova. But this is not happening in a massive star, but it's happening in an average star. So we start giving names like type 1A, type 1B. Like this, we'll give different names for this. So it's a detonation that happens in a star, but not in a massive star, but it happens in an average star. That's all. Is it clear? Massive stars. Type followed by any number, it happens in a smaller star, average star. Is there any doubts? Can you go to the next one? Right. We're starting with biology first before nuclear science. I thought I will give a basic introduction to biology. Followed by that, we will go into nuclear science. Right, plus biology. The biggest challenge in studying biology is you'll always have a question where to start. Because 6th to 12th standard, a lot of books are there. Where to exactly start reading biology? Let me take my notes. So where to exactly start reading biology? This will be the biggest question that you will have. But I would advise you, don't read it class-wise, read it topic-wise. For example, if you're starting with nutrition, or if you're starting with, diseases, health and hygiene. From 6th to 12th standard, wherever you find things related to health and hygiene, you have to cover it. Only if you cover topic-wise, you'll be able to complete it. But this basic introduction will give you a very good head start, so that when you read your NCERTs, it will be much easier. First, we are starting with classification of organisms. Put the title as classification of organisms. So the classification of organisms, because we are humans and then we have plants like this. There are different things. Now, how do we put it into a classification? There are different methods. Have you heard about kingdoms of classification and everything? We have these many methods of classification, but in this NCERT, or the most widely used one is given by Whitaker. We have 2 empire and 5 kingdom classification. We are going to learn about the 5 kingdom and 2 empire classification. But before that, you're going to get familiar with some of the terminologies. First is prokaryotes, and then you have eukaryotes. What are prokaryotes and what are eukaryotes? Any random guesses? Prokaryotes are often called as false nuclei. Eukaryotes are called as true nuclei. That is, under prokaryotes, you can write the cell organelles are not properly developed. Cell organelles are not properly developed for prokaryotes. Cell organelles are not properly developed. Prokaryotes. Cell organelles are not properly developed in prokaryotes. For eukaryotes, the reason we call them as true nuclei is cell organelles are properly developed. You can write cell organelles are properly developed. cell organelles are properly developed. Okay. This is one classification. Second classification is, you take that this is the cell of an organism. What is the outermost part of the cell? Generally called as cell wall, followed by the next one is cell membrane, otherwise called as the plasma membrane. Keep in mind, sir, for certain organisms, outermost part will be cell wall. For certain organisms, the outermost part will be cell membrane. So we have one more classification based on the outermost covering of the cell. It can be either cell wall or it can be cell membrane also. Is it clear? Okay, next one. Very familiar. Unicellular and then multicellular. What is mean by unicellular? Made up of single cell. Multicellular, made up of multiple cells. Optional, if you want to write, you can write. Unicellular or organisms made up of single cell. Multicellular or organisms made up of multiple cell. Yes, so far we have learned about prokaryotes and eukaryotes. Then about organisms with cell wall and without cell wall. And then unicellular organisms and then multicellular organisms. And based on modes of nutrition, we have autotrophic, heterotrophic. What is meant by autotrophic? Organisms that can prepare their own food. Those are called as autotrophic organisms. And heterotrophic depends on other organisms. For example, we are heterotrophic. Plants are autotrophic. Organisms capable of synthesizing their own food are called as autotrophic, and organisms that depend on other organisms for food are called as heterotrophic. Produces food on own, depends on other organisms for food. Autotrophic and heterotrophic. What is this? Now, I said 2 empires and 5 kingdoms, no? The NCERT would have given something like this, but we will simplify in this manner. See here, organisms are divided into 2 types. And there are 5 kingdoms: Monera, Protista, Plantae, Fungi, and Animalia. We'll talk about it one by one. See, what is the one given here? Prokaryote. What does it mean by prokaryote? Will not have proper cell organelles. Under prokaryote, do you see one? Monera. Everything that belongs to the Monera kingdom, the 5 kingdom. Monera kingdom is a prokaryote, which means none of these organisms will have properly developed nucleus. Under eukaryotes, you have 2, unicellular and multicellular. What is written under unicellular? Protista, which means every organism that belongs to the Protista kingdom will be made up of one cell, and that one cell will be properly developed. Under multicellular, with cell wall and without cell wall, you take fungi and plantae. Every organism that belongs to the fungi and plantae kingdom will have cell wall, and there are multiple cells, and all these cells are properly developed. And then you take animalia. We have an example for animalia. Animalia, for example, humans, they are made without cell wall, and they are made of multiple cells, and these cells are properly developed, which means we are multicellular eukaryotic organisms. And if you take euglena and everything, they are unicellular eukaryotic organisms. Bacteria, prokaryotic organisms. So this is the classification. First you write this. Then I will tell you example for Monera, Protista, Fungi, Plantae, Animalia, and everything. Noted this? Yes. We will write examples. Example for first kingdom, Monera. For Monera, I can write the bacteria or bacterium. Their example for Monera kingdom. Unicellular eukaryote. Then the Protista kingdom, you can write euglenoids, dinoflagellates, all these are examples for Protista. Fungi, most of the mushrooms we consume are agaricus. All these are examples for fungi kingdom. Plantae or plant kingdom, any plant that you like. Apple tree, mango tree, or hibiscus, anything can be taken as example. In Animalia kingdom, we humans are examples for Animalia kingdom, right? This is a general classification. Then, to the next one. You remember seeing this somewhere? Plant cell or animal cell? This is random guess. Plant or animal? Animal. This is plant cell. So if you compare, you'll easily understand. Maybe in standard, you would have seen this standard diagram for your public exams. Entire exam hall will draw this, and then you'll hear this. You put the cell organelles. This is animal cell, and this is a plant cell. We're going to compare and learn about all the organelles that are present here, because 2019 UPSC and 2020, they've asked about cell organelles. Just about the organelles present here. So protoplasm, cytoplasm, nucleus, these things you should have a clarity. You've heard about protoplasm, you've heard about cytoplasm. What is the difference between a protoplasm and cytoplasm? To understand this, if you take any cell, I've drawn this earlier, but still once again, which is the outermost part called as cell wall. And then the second one is cell membrane, or otherwise called as plasma membrane, followed by cell wall and cell membrane. This entire content that you see here, no, that will be called as protoplasm. So the entire content of the cell will be called as protoplasm. Then, which is the control center of the cell? Nucleus. Let's say this is the nucleus. Now, what we are going to do is, please observe here carefully, the entire content is called as protoplasm, correct? Then this is nucleus. I'm writing the control center is called as nucleus. If you take everything outside the nucleus, but within the plasma membrane, there is, do you see a disc-shaped portion in that purple color? That will be called as cytoplasm. That is, I will put it this way. Protoplasm equals to cytoplasm plus the nucleus. That is this is cytoplasm. This color is nucleus. Both of them joined together. The combined one is called as protoplasm. Is it clear? Only the disc-shaped portion is called as cytoplasm. This is called as nucleus. Cytoplasm plus nucleus. The entire package is called as protoplasm. Why we are in particular learning about protoplasm, cytoplasm, and everything is, when we learn about biotechnology, no, say we are talking about protein synthesis, we'll straight away say that template is copied from nucleus to the cytoplasm. You should understand what is nucleus and what is cytoplasm. Then only you will understand the biotech processes. So whatever you're learning in the biology will form the basic for the biotechnology concepts. Clear? First copy this protoplasm and cytoplasm. Can we go to the next one? Right. I will send you the mind map later. See what is written here in cell organelles. You have cell wall and cell membrane, which are not part of protoplasm. In protoplasm, what are the two things we have written? Cytoplasm and nucleus. In cytoplasm, you have these many cell organelles. In nucleus, you have these many organelles. We are going to learn about one by one. Clear? So if you see this, this is the nucleus, and whatever you find here will be cytoplasm. Together it is called as protoplasm. Once again, this entire content will be called as what? Protoplasm. And then you are leaving the nucleus, and then the highlighted portion will be called as cytoplasm. Have you understood this? Okay, first organelle that we are going to talk about is cell wall. What the title is? Cell wall. Okay. The cell wall first, it gives shape and structure to the cells. Cell wall gives shape and structure to the cells. Cell wall gives shape and structure to the cells. Next line you can write, it protects the cell from damage. It protects the cell from damage. Cell wall protects the cell from damage. Cell wall is present in plant cells, absent in animal cells. Cell wall present in plant cells and it's absent in animal cells. Okay, what's the role of cell wall? Protects the cell, gives the shape, gives the structure. Cell walls are made up of familiar statement. Cell wall is made up of cellulose. This is what you would have studied in school time usually, but not necessarily cellulose always. Plant cells are made up of cellulose. But if you take fungi, cell wall may be made of materials like chitin. So generally cellulose, chitin, if you see any of these terms associated with cell wall. Example for materials that make cell wall, cellulose or chitin. Right. Next one is cell membrane or plasma membrane. Next is cell membrane or plasma membrane. First line you can write, it's a semi-permeable membrane. Why we call it a semi-permeable? Because it allows certain things, it will stop certain things. That's why it's called a semi-permeable. So what enters or leaves the cell, it's decided by the cell membrane. It's more like a barrier. Sometimes it will allow things, sometimes it will stop things. It's responsible for both active and passive transfer. It's responsible for active as well as passive transfer. When we say active transfer, it generally needs energy, and passive transfer, it happens automatically because of change in concentrations. Active and passive transfer or transport. You've heard about osmosis and everything, no? If you put a resin or something inside water, the water is imbibed. All these are passive. Because of the change in concentration, it's automatically happening. But if you want to do it in reverse direction, reverse osmosis, for example, you need to put power. So active transfer and passive transfer, or active transport and passive transport. Active transport needs energy. Passive transport will not require input of energy. It happens automatically. Basically, all these happens through this. That is semi-permeable membrane called as plasma membrane. You've completed cell wall and cell membrane. The next one is vacuoles. Next is vacuoles. What does it mean by a vacuole generally? Gap or space is called as a vacuole. You see this picture, you take plant cell and animal cell. This is animal cell, this is plant cell. Which has huge gap? Plant cell. Can you see here? There's large gaps here. These are called as vacuoles. Vacuoles are generally larger in plant cell and very smaller in animal cells. You can write vacuoles are storage sacs. Vacuoles are storage sacs. When we say storage, it stores water, sap, or other nutrients will be stored in vacuoles. And vacuoles are larger in plant cell and they are smaller in animal cells. Vacuoles, they are larger in plant cells and smaller in animal cells. Can you go to the next? Next is endoplasmic reticulum. Endoplasmic reticulum. The endoplasmic reticulum can be divided into two types. One is smooth endoplasmic reticulum. The other one is rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. Smooth endoplasmic reticulum. Rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and rough endoplasmic reticulum. We are dividing into two, smooth and rough. Smooth and ro Why we call it as smooth, why we call it as rough. The picture that you see here, this is the endoplasmic reticulum that extends from the nucleus. On this endoplasmic reticulum, do you see certain granules attached, which gives a rough appearance? Those are ribosomes. That's called as a rough endoplasmic reticulum. And then in the bottom, do you see this? The same folded structure, but here you do not have any particles on the top. So these are called as smooth endoplasmic reticulum. The difference between smooth and rough endoplasmic reticulum is, for smooth endoplasmic reticulum, and ribosomes are absent. Ribosomes are the granular particles. Rough endoplasmic reticulum, ribosomes are present. Why ribosomes are important? Next, any idea what is the role of smooth and rough endoplasmic reticulum in general? Smooth endoplasmic reticulum are related to lipid synthesis in the human body. When we say lipid synthesis, but fat, all these falls under lipid synthesis. Butter, for example, it's rich in lipids. So lipid synthesis includes fat, cholesterol, and everything. It's guided by smooth endoplasmic reticulum. Coming to rough endoplasmic reticulum, it's related to protein synthesis. Rough endoplasmic reticulum is related to protein synthesis. Out of this, again, close the tabular column. We completed two types of endoplasmic reticulum. Next is Golgi apparatus. Golgi apparatus. So what's this Golgi apparatus? When something is produced in the cell, and then it has to be sent across the cell to different cells or cellular vesicles, you have to pack them, correct? That is performed by Golgi apparatus. Golgi apparatus is more like a logistic partner. It will pack it in a particular form and then it will send it across the body. You can write three important terms. First is storage, modification, and packing. Storage, modification, and packing. These are the three important functions of Golgi apparatus. First, it stores food, and then it will modify it in a particular form, and then it will pack and send it across the cellular vesicles. So in general, you remember, packing and everything is performed by Golgi apparatus. Next one is lysosomes.